Hello everyone, today we'll talk about lecture 5, alternative molecules, alternative translation initiation. This is lecture number 3 in the alternative molecules, and this is lecture number 5 in the course Molecular Genetics MLGN 301 2006. I hope you like this new lecture, which complete the 3 set of lectures about alternative molecules. In this lecture we'll talk about alternative translational initiation, and as we said, there's some recommended book for further information, like the book Bioinformatics: A Practical Approach. In this lecture we hope to give some outcomes for the student, the learning and understanding. Number one, what is alternative translation initiation, why alternative translation initiation is important, and of course the student should know some examples of alternative translation, understand a difference between global and alternative translation initiation. As we see, in a normal translation means that the DNA will transcribed into RNA, this inside the nucleus, and of course RNA should go outside the nucleus to the cytoplasm and translated to a protein. So the transformation from DNA to RNA is called transcription, and this happened inside the nucleus, and the transformation of RNA into a protein, this called a translation, and this happened outside the nucleus membrane, inside the cytoplasm, and of course with the help of the ribosome. As we can see that the ribosome will read the messenger RNA, and this will be the messenger RNA will be translated into a protein via the ribosome. The ribosome will collect all the transfer RNA which have the amino acids which represented in a code inside the mRNA, and then they build the protein as we know in a normal cell. So the translation maps of RNA to a protein from 3-letter code to a 1-letter code. As we see that always the 3-letter code in the messenger RNA, every 3-letter correspond to 1 amino acid. Every 3-letter in the messenger RNA correspond to 1 amino acid that build the chain of amino acids, the polypeptide chain, which will be after that will be a part of the protein. So that happens inside the ribosome. So the messenger RNA attached to the ribosome, and then the ribosome read every 3 codes and translate it to 1 code. In eukaryotic cell, translation is usually initiated according to the ribosome scanning model. This is called ribosome scanning model. Now, what is ribosome scanning model? This is the 40S ribosomal subunit, which is a small subunit, and the translation initiation factor bind to the 5' end of the messenger RNA and scan the RNA molecules in the 3' direction until they reach AUG codon. So as we see, the smaller part of the ribosome attached to the 5' and then scan until they reach the begin, and then start then the 80S ribosome assembles and begin protein synthesis. So this is a smaller part. First it attach, and then until attached to the messenger RNA, and then until they reach the AUG site, and then the bigger part, part of the ribosome, 60S or 80S, whatever, assembles, and this start the translation of this messenger RNA. This ribosomal complex start translation, and the initiation of translation start. So start always in the AUG. So this is the normal translation of the messenger RNA. So what about the alternative translation? This means that there is another way for the translation and an alternative way. So the normal translation is that the smaller ribosome unit will scanning model, and then this scanning model go until it reach the AUG, and when it reach the AUG, the bigger ribosomal part, 60S or 80S depending on the organism, and then this will be attached in the initiation of the translation start from here. So what is alternative translation initiation? So this means that this translation initiation model could be different. ATI, alternative translation initiation, is one of the mechanisms that increases the complexity level of the organism by alternative gene expression pathway. The use of ATI codons in a single messenger RNA contributes to the generation of protein diversity. So this is another mechanism that contribute to generation of protein diversity. So we have studied before other mechanism, but alternative translational initiation contributes to the protein diversity. So that's why we could have one gene, but this one messenger RNA, and this one messenger RNA can produce different proteins because of the alternative translational initiation. Or of course, as we see, the complexity could be increased, as we know that the genome contains from 20 to 25 genes, and we know that there is something we have studied alternative promoters before, and alternative promoter can generate more than 100,000 transcripts, and then the post-translational modification can contribute to many proteins, over 1 million different proteins. So this 1 million proteins can be generated from only 20 to 25,000 genes, and this what we call it proteome complexity. So anyway, so that alternative translational initiation contributes to this proteome complexity. So what is alternative translation initiation? The gene produce 2 or more versions of encoded protein, and the shorter version initiates from the downstream in-frame start codon, lacks the N-terminal. It's always like the N-terminal amino acids fragment of the full-length isoform version. So always we can see that the smaller version lacks the N-terminal of the full-length isoform version. So this means that the same messenger RNA can produce many isoform of the same protein, many isoform. Of course, this isoform could have different function or lose function. So as we can see that the initiation could start here or here or here or there. All these points can be a point of initiation for translation for the same messenger RNA, depending on the when and how and where this translation happen. So this initiation, when it's because the diversity of this initiation could produce diverse product. This of course have a physiological and pathological implication, because the structure feature within the 5' UTR and translation, such as the secondary structure and operating frame, can strongly influence the efficiency of translational initiation. And this of course means that you will have a different isoform of the protein, which means that this protein could have a different function or lack of function. So this means that it will have a physiological and pathological implication. And of course there is many examples of that. Let's see, for example, for biological significance, we have the example of bFGF, basic fibroblast growth factor. There is several forms of bFGF, basic fibroblast growth factor, are detected in the most producing cell types, and these different forms are result from alternative initiation of translation at the EUG codon at the 3' frame upstream CUG codon, leading respectively to synthesis of a small form which is 19 kilodalton, or a large forms like 21, 21.1, 21.5 and 22.5 kilodalton isoform. So we have the same gene produce the same messenger RNA, and the same messenger RNA can be translated alternatively by different ways, and the same messenger RNA can produce different isoform of protein and in different cell types, and as we see in the example of bFGF gene. The expression of AUG initiated from 18 kilodalton leads to cell transformation, where expression of the CUG initiated form leads to cell immortalization. So even the function of this isoform are different. So we have 18 kilodalton leads to cell transformation. Other expression of another forms leads to immortalization of the cell. So this means that we have different the same gene produce the same messenger RNA, which the same messenger RNA produce different isoform of protein, and every protein can go to different cell types and leads to a different function. In the translation of the bFGF messenger RNA, 5 cis-acting elements located in the 5' UTR of the bFGF messenger RNA are able to modulate the global and alternative use of 4 initiation codon. And we have to know the difference between global and alternative. So global means the total length, the full length of the translation of the messenger RNA, and alternative means not the full length of the messenger RNA. So here the control of the alternative initiation of translation of bFGF messenger RNA have an important impact on the cell behavior. So this means that this isoform can affect the cell behavior. There is, of course, many other examples like this receptor gene and its product, and many other genes in this regard, and you can read across all this activity of that different genes, and there is several other examples can be described like LBPM5 immunovirus, murine leukemia virus, and ATRX alpha thalassemia syndrome, and foot and mouth disease. All these examples are examples of this ATI topic. And of course, because there is many other examples, we have actually a database for that. But anyway, as you can see that this, for example, this gene, that you can have a product which is original product or the full length product, and you have isoform. So this, when we call about protein isoform, you have a full length protein, and you have other product that is have less size. And less size means that the number of amino acids are less. So that's why you can see that the start here from 1 to 288, and here's the start of from the amino acid number 79, and the end of 288 also, and here the start is in the 88 amino acid number 88, and then go to the end, and like this. So the last version is a smaller version, isoform, which lack much more amino acids. It's the smaller version, and the full version starts from the first amino acid number 1, and the end is in the end 288. For example, this is an example for what the meaning of alternative isoform of the same protein. Of course, as we said, there is many examples, and there is because there is many example, we have a database on translational initiation, and this database can tell you if your protein have different isoform or not. So simply this is how the database looks like. You can just click on this database and take a look, and of course you can search for whatever gene you want, whatever protein you would like to see. You can just put the name of the gene, and just put it here, and or put the sequence and of the nucleotides or amino acid, and then click search. This will give you a list of results that mean that Imporus taurus, for example, have this gene, but this gene have 2 isoform, and the Gallus have 2 isoform from the same gene. Homo sapiens have 2 isoform from the same gene. So simply this database can tell you if your protein of your gene have a different isoform of the protein or not. As we just mentioned, that this genome complexity is increased by alternative promoter, alternative splicing, and alternative translational initiation, and this can lead to what we call proteome complexity, which means that the number of the genome genes in the genome can be increased dramatically. This is a practical. What is alternative translation initiation? Why alternative translation initiation is important? Describe 2 examples of alternative translation initiation. What are the differences between global and alternative translational initiation? Thank you for listening, and I hope to follow us in the channel, in the Alzohairy channel on the YouTube, and see more videos. And please comment, like, and subscribe. Thank you.